Platelet activating factor. It is released from the IgE sensitized basophils or mast cells, other leukocytes, endothelium, and platelet. Apart from its action on platelet aggregation, release reaction, the action of platelet activating factor. So first action is aggregation and release reaction, and some other actions are mediator of inflammations like increased vascular permeability, vasodilation in low concentration, and vasoconstriction otherwise. Bronchoconstriction, adhesion of leukocytes to endothelium, and chemotaxis. Now let's see cytokines. Cytokines are polypeptide substances produced by activated lymphocytes, lymphokines, and activated monocytes, monokine. The term chemokine is used for a family of substances which can act as chemoattractant for inflammatory cell. All these agents can act as self cells which produce them or on the other cells. Although over 200 cytokines have been described, major cytokines and their roles in the inflammation are described under the given table. So chemokines are basically polypeptide substances like kinins, but they are not similar. They are produced by lymphocytes. Remember this, who produce them. Then they would be known as lymphokines, and if they are produced by monocytes, then they would be known as monokines. The term chemokine is used for a family of substances which act as a chemoattractants for inflammatory cells. Now let us see the table 5.3. Major cytokines in inflammation. First is interleukin 1, which is major factor responsible for fever. So the cell source is monocyte, macrophage, B cells and fibroblast, endothelial cells and some epithelial cells. Cell target is all cell, so the interleukin can act on all the cells, and the main action is expression of adhesion molecules, emigration of neutrophils and macrophage, role in fever and shock, hepatic production of acute phase reaction. So let us repeat. First is interleukin 1, which is produced from the monocyte or macrophage, B cell, fibroblast, endothelial cells and some epithelial cells. It acts on all the cells and is responsible for expression of adhesion molecules, emigration of neutrophil and macrophages, helps in fever and shock production, and hepatic production of acute phase reaction. Now let's see interleukin 6. Same source as those of interleukin 1, which are monocyte or macrophage, B cells, fibroblast, endothelial cells and some epithelial cells. Target cell are B and T cells and epithelial cells, hepatocytes, monocytes or macrophage. Main action is hepatic production of acute phase protein and differentiation and growth of B and T cells. Now let us see. So in the IL-6, the target cells are those which produce the same. So it can be called as autocrine. Now IL-8, monocyte or macrophage, T cells, neutrophils, fibroblasts, endothelial cells and epithelial cells. Target cell is neutrophil, basophil, T cell, monocyte or macrophage and endothelial cell. Main action is induces migration of neutrophil, macrophage and T cells, stimulates the release of histamine from the basophil and stimulates the angiogenesis. Then IL-12 macrophage, dendritic cell and neutrophils, acts on T cells and NK cells, and action is induces formation of helper T cells and killer cells, promotes CTL cytolytic activity, increases the production of INF gamma and decreases the production of IL-17. Then IL-17, CD4 and T cells, fibroblast, endothelial cells and epithelial cells, increases the secretion of other cytokines and migration of neutrophils and monocytes. TNF alpha, the monocytes and macrophage, mast cells or basophils, eosinophils, B cells and T cells and neurocrine cells. Target cell is all the cell except RBC. Mechanism of action is hepatic production of acute phase protein. Systemic features are fever, shock and anorexia. Expression of endothelial cell adhesion molecules enhances the leukocyte cytotoxicity, induces induction of pro-inflammatory cytokines. Now INF gamma, cell source is T cells and NK cells. Target cell is all the cells. Activation of macrophage and NK cells. Stimulate the secretion of Igs by B cells. Differentiation of helper T cells. MCP1, fibroblast, smooth muscle cells, blood mononuclear cells. Target cell is monocyte, macrophage, NK cells and T cells. Action is chemoattractant for monocyte, T cell and NK cells. Stimulate the release of histamine from basophils. Eotaxin, cell source is alveolar cells and myocardium. Target cell is eosinophils and basophils. Act as a chemoattractant for eosinophil and basophil and induces the allergic pulmonary diseases. Then we have PF4, which is source is platelets and megakaryocytes and acts on fibroblast and endothelial cells. Chemoattractant for fibroblast, inhibitory to hematopoietic precursor and endothelial cell proliferation. Interleukins IL, TNF is tumor necrosis factor, IFN is interferons, MCP is monocyte chemotactic protein and PF is platelet factor. Now let us see the interleukins. Interleukin 1, 6, 8, 12 and 17. While interleukin 1 and 6 are active in mediating acute inflammation, interleukin 12 and 17 plays a role in chronic inflammation. So this is important, interleukin 1 and 6 for acute inflammation and interleukin 12 and 17 for chronic inflammation. The interleukin 8 is chemokine for acute inflammatory cells. Interleukin 1 is elaborated by several body cell monocytes and macrophages, B lymphocyte, fibroblasts, endothelial and some epithelial cells. Similarly, it can target all body cells. Its major action are expression of adhesion molecules, emigration of neutrophil and macrophages, role in fever and shock, hepatic production of acute phase protein. Interleukin 6 is similar in its source and target cell of action. Its major role are hepatic production of acute phase protein and differentiation and growth of T and B cells. Interleukin 8 is also elaborated by same cells as for interleukin 2 and interleukin 6, except that It is secreted by T cells instead of B lymphocytes. Its target cells are neutrophil, basophil, T cells, monocyte, macrophage, endothelial cells. IL-8 is a chemokine and its major actions are induces the margination of neutrophil, macrophages and T cells, stimulate the release of histamine from basophils and stimulates the angiogenesis. Interleukin 12 is synthesized by macrophage, dendritic cells and neutrophil while it targets T cells and NK cells. Its major action is chronic inflammation are as under. Induces the formation of T helper and killer cells, promotes the CTL cytolytic activity, increases the production of INF gamma and decreases the production of interleukin 17. Interleukin 17 is formed by CD4 and T cells while it targets fibroblasts, endothelial cells and epithelial cells. Its action is chronic inflammation are increased secretion of other cytokines, margination of neutrophils and monocytes. Tumor necrosis factor alpha and beta. Alpha mediator of acute inflammation while beta is involved in cytotoxicity and in the development of spleen and lymph nodes. TNF alpha is formed by various cells, monocytes, macrophage, mast cells or basophils, eosinophils, B cells, T cells and NK cells, while TNF beta is formed by B and T cells. Both can target all the cells except erythrocyte. Major action of TNF alpha are hepatic production of acute phase protein. Systemic features are fever, shock and anorexia. Expression of endothelial adhesion molecules enhances the leukocyte cytotoxicity. Production of pro-inflammatory cytokines. Interferons gamma. It is produced by T cells and NK cells and may act on all body cells. It acts as a mediator of acute inflammation as under. Activities of macrophage and NK cells stimulate the secretion of immunoglobulins by B cells and role in differentiation of T helper cells. Other chemokine like IL-8, MCP1, eotaxin, PF4. Besides the IL-8, a few other chemoattractants for various cells are as under. MCP1 elaborated by fibroblasts, smooth muscle cells and peripheral blood mononuclear cells. Its action are chemoattractant for monocyte, T cell and NK cells and stimulate the release of histamine from basophils. Eotaxin is formed by alveolar cell of the lung and in the heart. Its action are chemoattractant for eosinophil and basophil and induces the allergic pulmonary disease. PF4 is formed by platelets and megakaryocytes and may act on fibroblast and endothelial cells. So the action of PF4 are chemoattractant for fibroblasts and inhibitory to hemopoietic precursor and angiogenesis. 6. Free radical oxygen metabolites and nitric oxide. Free radicals act as a potent mediator of inflammation. First, oxygen-derived metabolites are released from activated neutrophils and macrophages and include superoxide, oxygen, hydrogen peroxide, hydroxide and toxic nitric oxide products. These oxygen-derived free radicals have the following action in inflammation: endothelial cell damage and thereby increased vascular permeability, activation of protease and inactivation of anti-protease, causing tissue matrix damage to other cells. The action of free radicals are counteracted by antitoxins present in the cell and serum, which play a protective role. Nitric oxide was originally described as vascular relaxant factor produced by endothelial cells. Now it is known as nitric oxide is formed by activated macrophage during oxidation of arginine by the action of enzyme nitric oxide synthase. Nitric oxide plays the role in mediating the inflammation, vasodilation, active antiplatelet activating agent, possibly microbial action. Plasma protein derived mediator, plasma protease. These include various products derived from the interaction of four interlinked systems: kinin, clotting, fibrinolytic, and complement. Each of these has its inhibitors and accelerators in the plasma with negative and positive feedback mechanism respectively. Hagman factor 12 of clotting system plays a key role in interaction of the four systems. So this can be asked in the MCQ, that which factors plays a role in interaction of the all four systems of plasma protein derived mediators. Then it is clotting factor number 12, that is Hagman factor. Activation of factor 12 in vivo by contact with the basement membrane and bacterial endotoxins, and in vitro activation with glass or kaolin leads to activation of clotting, fibrinolytic, and kinin system altogether. In inflammation, activation of factor 12 is brought about by contact of the factor leaking through the endothelial gaps. The end product of activated clotting factor and kinin system activate the complement system that generate the permeability factor. This permeability factor in turn further activate the clotting system. The interrelationship among the four systems is summarized in this figure. Factor 12 contact factor 12 will be activated, which leads to fibrinolytic system activation, which releases the plasmin. Activation of clotting system, where the fibrin is acted upon by plasmin, which leads to fibrin split products, and which will activate the complement system. And there will also be activation of kinin system, which leads to kallikrein and bradykinin, which all these will activate the vessel wall and leads to permeability factor activation C3A, C5A, and MAC, C5B to C9. Now let us continue with the kinin system. The kinin system, this system on activation by factor 12A generates bradykinin, so named because of the slow contraction of smooth muscle induced by it. First, kallikrein is formed from the plasma pre-kallikrein by the action of p-kallikrein activator, which is a fragment of factor 12A. Kallikrein then acts on high molecular weight kininogen to form bradykinin. Bradykinin acts in the early stage of inflammation, and its effects include smooth muscle contraction, vasodilation, increased vascular permeability, and pain. Second is the clotting system. Factor activated 12 initiates the cascade of clotting system resulting in the formation of fibrinogen, which is acted upon by the thrombin to form fibrin. Fibrinopeptides are given in figure 5.12. So, activated factor 12 will leads to activation of factor 11, then 8, then PF3, which all of three together activates the factor 10, 5 and PF3, which combinedly activates the thrombin from prothrombin, which activates the fibrin from fibrinogen, which is acted upon by the plasmin, which leads to formation of fibrinopeptides. Now let us see the actions of fibrinopeptide in inflammation: increased vascular permeability, chemotaxis for leukocytes, and anticoagulant activity. Third, the fibrinolytic system. This system is activated by plasminogen activator, the source of which includes kallikrein of the kinin system, endothelial cells, leukocytes. Plasminogen activator acts on the plasminogen present as the component of plasma protein to form the plasmin. Further, breakdown of fibrinogen by plasmin forms fibrinopeptides or fibrin split products. The action of plasmin in inflammation are as follows. First, activation of factor 12 to form the prekallikrein activator that stimulates the kinin system to generate bradykinin. This is a bit tricky, so again, the action of plasmin in inflammation are as follows. Activation of factor 12 to form prekallikrein activator that stimulates the kinin system to generate bradykinin. So first there is production of bradykinin, then split of complement C3 to form C3A, which is permeability factor, and degrades the fibrin to form fibrin split product, which increase the vascular permeability and are chemotactic to leukocytes. So as the factor 12 activates prekallikrein activator, plasma prekallikrein is converted to kallikrein, which acts on kininogen to form bradykinin. Fourth is the complement system. The activation of complement system can occur either by classical pathway through antigen-antibody complexes or by alternate pathway via non-immunological agent such as bacterial toxins, cobra venom and IgA. So basically complement system activation can either occur by antigen-antibody reactions or by non-immunogenic agents like bacterial toxins or cobra venom or IgA. Complement system or activation by either of these two pathways yields activated product which includes anaphylactones, phylactoxins like C3A, C34 and C5A. C3A, C4A and C5A, and the membrane attack complex, that is C5B, C6, C7, C8 and C9. The action of activated complement system in inflammation are as under. C3A, 5A and 4A anaphylactoxins activate the mast cells and basophils to release histamine and cause increased vascular permeability, causing edema in the tissue, augments the phagocytosis. So what does C3A, 5A and 4A will do? They will activate mast cell and basophil to release the histamine, which will increase the vascular permeability, which leads to movement of the proteins into the extravascular space in the tissues, which leads to edema in tissues and augments the phagocytosis. C3b is an opsonin. C5a is a chemotactic for leukocytes. Membrane attack complex MAC is a lipid dissolving agent and causes holes in the phospholipid membrane of the cell. So these functions of the complement system, and the whole complement system is very important. So repeating again, the activation of complement system can either occur by classical pathway through antigen antibody complexes or by alternate pathway via non-immunologic agent such as bacterial toxin, cobra venom and IgA. So there are two ways for activation of complement system. Upon activation, activated product like anaphylactoxins, C3A, C4A and C5A and membrane attack complexes like C5B, C6, C7, C8 and C9. The action of activated complement system in inflammation are as under: C3A, 5A and 4A. Anaphylactoxin activates the mast cell and basophils to release the histamine, which cause increased vascular permeability, causing edema in tissue and augments the phagocytosis, whereas the membrane attack complexes like C3b is an opsonin, C5a is a chemotactic for leukocytes, and membrane attack complex is a lipid dissolving agent and causes holes in the, so it attacks membrane by causing holes in the phospholipid membrane of the cell. From regulation of the inflammation, we will continue in the next video.